Here we see striped ones and unstriped ones and long ones and thin ones. And one has to ask the question, why is there this variation?
We're right down to 3 adults. These are the only ones left in the world.
You're looking at a species which has evolved to fit beautifully in its own niche.
There's a whole different world of smaller animals. They're just wonderful, wonderful things.
These 4 people have struggled to prevent a genocide. If they succeed, they could not only solve some great evolutionary puzzles, they will save from extinction an entire family of snails called Partula. Curiously, the force that threatens Partula is another snail. Last year, every night for a fortnight, sleep was sacrificed to science. A team of committed zoologists spent their evenings in the jungle of the palm house at Kew, crawling around in the undergrowth in search of snails. These snail experts are using Kew to build a model of how Partula may behave in their wild environment. It's all part of their fight to save the animal that has become the focus of their careers.
What we've done is to release a group of snails from the captive breeding program onto this large pandanus with the intention of seeing how the long-term captive bred snails readapt to being on native plants again.
They're actually mainly nocturnal. They actually like moving around when it's very wet, but particularly at night. That's why we're here with the reddened light, so the animals can't actually get disturbed by bright light. And we've got special radioactive markers on the back of the shells, and there's an adult here, number 21, and it makes it a lot easier to spot the animals at night.
The object of all this attention is a peaceful tree-dwelling snail. There are 123 different species of Partula, all but one are vegetarian. But quite apart from this unassuming creature's right to life, there is another reason for saving Partula. They have been the focus of some of the most significant evolutionary research this century. Not so much because of how they live, as where. In the geographical center of the Pacific lies the island of Moorea. It is home to 7 different species of Partula and a second home to Professor Brian Clark.
If you're interested in evolution, you have to study islands almost inevitably. The wonderful thing about them is that volcanic islands pop up out of the sea from time to time, and there they are new. When an island pops up, it's got nothing on it. Then it becomes populated firstly by plants, and then some animals get onto it, and they find this wonderfully diverse, rich environment with nothing much else in it. And so then they diversify like mad. Evolution starts rampaging, and they differentiate into lots of different varieties and species.
From Darwin's time, generations of biologists have recognized that islands are living laboratories of evolution, and Moorea is one of the finest. From its volcanic mountaintops to the seashore, the island has an extraordinary range of environments. And it was to these virgin biological niches about 1 million years ago that the tiny founding population of Partula were carried on the feet of birds. They arrived, dispersed, and evolved.
And what stimulated our interest in this process was this monograph by Crampton, Henry Edward Crampton, who was a professor at Columbia. And he studied the Partula that live on the island of Moorea and produced wonderful descriptions and these pictures of all the varieties and all the species, which enabled us to have a really good start in studying the evolutionary process.
This monograph, a catalogue of 116,000 snails, is one of the seminal texts of early population genetics. Henry Edward Crampton first went to Moorea in 1902 and believed his work on the island, like Darwin's on the Galapagos, might help explain how species evolve. Inspired by this epic work, in 1962, Brian Clark, in partnership with Jim Murray from the University of Virginia, followed in Crampton's footsteps. They too were captivated by Partula.
People are liable to burst into fits of laughter when you say you study snails, or to make remarks like you should have no difficulty in keeping up with your subject. But they have really very many advantages if you're a student of evolution. If you want to observe populations and how they change and evolve, you want something that when you look at it one year, you'll know it'll be there again the next, i.e. you want something with low mobility. Now, the other advantage of snails is they've got shells, and therefore you can mark individuals and you can study their fate. And the third thing is they live on shrubs, and so you can collect them. These things made Partula a really wonderful organism for studying the relationship between the organism and its environment and how this relationship evolves.
This study of Partula would be going on today, except that in the late 1960s, everything suddenly changed. Polynesia, first settled by great seafaring explorers, has always had migrants. New arrivals, and with them, new foodstuffs. The waves of migration are reflected in the language and in the faces of the people. This constant influx has transformed the island's original ecosystem. These biological imports have escaped their native system of checks and balances. Some plants and animals introduced as produce have now become pests. Unlike the native Partula, these edible snails were brought here by man. Their arrival had a dramatic effect.
Billions and billions have already eaten every blade of grass and all the jungle growth on scores of islands. The pests appeared in vast numbers one summer morning, and by nightfall, several thousand acres of farmland were literally crawling with the creatures. Next day, not a live snail could be found on the island. It's guessed that the snails, impelled by some strange urge, committed mass suicide in the sea.
The snail with the strange suicidal urge was Achatina fulica, the giant African land snail.
When the body comes out of the shell, it can reach a length in excess of 30 centimeters, so they're absolutely huge. And an individual this size could weigh as much as 1 kilogram.
These mighty mollusks started to move during the 19th century, traveling slowly from their East African home, heading inexorably for Polynesia. Transport wasn't a problem. It's said they were even carried unconsciously on humans. During the Second World War, the land snail speeded up. It spread to many of the Pacific islands. Rumor has it Japanese soldiers introduced the snails out of sheer spite. The fact is, they introduced them to eat. Achatinas were washed in at least 2 changes of water, gutted and eaten with soy sauce. A breakfast of snail soup was a particular favorite. The war ended, but even the resumption of normal life could not halt the land snail's advance.
This is probably the most important book ever written on the African land snail. It was written by Professor Al Mead back in 1961, and one of the things which Professor Mead highlights is this species' in prodigious reproductive potential.
By 1967, this phenomenal breeder had reached Tahiti. According to local gossip, the French governor here imported them as a culinary delight. Certainly Chef Gaspard du Rollet thought them excellent. But it was an invasion that ultimately threatened Partula.
Now, if I could just make a quick quote from Mead, at the end of a 3-year period, all of the progeny from one fertile individual, if they all survived, could exceed 8 billion snails.
On Moorea, with no natural predators, most of their progeny did survive.
L'escargot géant d'Afrique est arrivé à Tahiti et environ un an et demi après, nous les avons trouvés également à Moorea. Au début, on en voyait un peu, mais peu de temps après, quelques mois après, on en a vu des milliers et des millions. Il y en avait partout, partout. C'est bien simple, voyez-vous cette orangeraie qui est couverte d'herbe, il n'y avait plus un brin d'herbe dans cette plantation. Toute l'herbe avait disparu et la terre était complètement nue.
Action was required, but it would be action that threatened more than the African land snail.
Over the last 50 years or so, a wide range of control measures have been attempted to keep in check these large population build-ups. These have ranged from straightforward collecting the snails to flamethrowers, traps, chemical sprays, and of course biological control.
Biological control is one of the safe methods available in the general control techniques. It is fairly safe and economical, long-lasting. Once if it is established in a place, it automatically it works. You know, in the other control methods like insecticides, you have to repeat that one periodically.
Dr. Munyapin's gospel of biological control was embraced by departments of agriculture throughout the Pacific. And Moorea's farmers, their crops threatened, were ready to embrace it too.
Dans le territoire, il y avait tellement d'escargots que j'ai eu l'idée à ce moment-là de contacter le service de l'économie rurale de Papeete, à Tahiti, qui eux m'ont conseillé de faire venir le prédateur du géant d'Afrique. Alors, ils ont donc commandé pour moi une centaine de prédateurs que, à l'époque, je me rappelle, nous avions payé $1 demi-dollar pièce.
The agent of biological control Monsieur Nardi bought was also a snail, Euglandina rosea. A native of Florida, this voracious carnivore came with Dr. Munyapin's blessing, but in the African land snail, it had met its match.
It's the exocet missile of the snail world. They really do zero in on their prey. But there are some problems with it. They only eat up to 200 snails a year, and the giant land snail produces over 600 babies a year.
Yeah, and it's also very important to make this size comparison point. These things grow up very, very fast, and it's been shown very clearly that this predator much prefers to take the whole snail because it needs the shell for its own shell formation. It needs that calcium.
Although not averse to the flesh of the African land snail, the predator couldn't stomach its giant shell. So for a balanced diet, it tended to look elsewhere. And with 123 different species of bite-sized Partula available, it didn't have to look far.
I couldn't believe it. About 1980, we suddenly noticed the appearance of these Euglandina, these carnivores. And in the areas where the Euglandina occurred, there were none of our snails. And at the end of 4 or 5 years, there were no more Partulas on the whole island. We'd worked on this fantastic island for 20 years, and it had shown us all kinds of interesting evolutionary phenomena. This was one of our major study areas, and it had 4 species of Partula in it, and it yielded a lot of very interesting results. Now, if you go and look at it, you will find nothing except Achatina, the giant African snail, Euglandina, the carnivore, and then a lot of dead Partula shells.
They never really stood a chance, for Euglandina moved 10 times as fast as Partula. With the extra-sensitive chemoreceptors on the sides of their heads, they latch on to the wide, clear trails other snails leave behind. They will do one of 3 things with whatever they find at the end of the trail: ignore it, mate with it, or eat it. If it happens to be a Partula, they will turn it over and with the vast lips typical of predatory snails, eat first the body, and then if they can, the shell.
Wherever Euglandina were introduced, there's no question that large numbers of endemic snails were absolutely decimated. In the case of the Partulas on the Society Islands, they were only saved from the similar total fate because of the fact they were being researched from the genetic point of view.
For by the time Euglandina appeared in Polynesia, some Partula were safe and sound in lunchboxes. These were the lucky few, cared for in Nottingham by Vivian Frame.
These snails are Partula mirabilis. In actual fact, they're the only ones in the world. We now have 35 adults and about 55 babies. This wasn't always the case. At one time, we were right down to 3 adults, and things were quite scary. We gave them lots of tender loving care and also added a supplement to the diet, vitamin E. And this seemed to work the trick, and things are looking very well.
But what was it that had made Partula worthy of all this love and attention?
Well, the central question in evolution is why is there so much variation? Why is there so much variation within species? Why do you and I differ? Why do some people have inherited diseases? And why is the variation between species? Why are we different from the apes? So if you're trying to tackle variation, then of course the studies of snails on islands becomes immediately important because we can see this diversity in the process of being born.
On Moorea, an aspect of diversity that particularly intrigued Brian and Jim was the so-called handedness of different species of Partula, whether their shells coiled to the left or to the right. They believed the explanation for this would contribute to the great debate about the role of natural selection in evolution.
In this general area of Moorea, one tends to find 4 species of Partula. There are 2 small right-handed species, Partula tenuata and Partula mirabilis. There's also a large left-handed species, Partula tohiviana, and the 4th species is Partula suturralis, which is unusual in that there are individuals that coil either to the right or to the left. This is a sufficiently curious evolutionary situation that we really wanted to try to find out whether these different coils of snails occurred at random or whether there was something systematic in the environment that was responsible.
This involved finding out what vegetation each species was living on and also pinpointing their exact geographical location.
Wouldn't like you falling out of your tree.
Brian Clark admits this part of the research might not have been done without Jim Murray's enthusiasm for taking accurate compass bearings.
Okay, Brian, I can get a pretty good round of peaks from here. Okie doke. In the south, I have the notch on Tamaratofa. It's bearing...
They found that Partula suturalis from the south of the island coiled to the right, whereas in the north they coiled to the left. Yet the vegetation they lived on was the same. So as this could not account for the coil of the suturalis shells, they decided to look at the snail's neighbors.
The next stage was to look at the other species of Partula that accompanied Suturalis, and what we found was that the nearest relative of Suturalis almost always coils in the opposite direction.
Since snails with opposite coiling shells find it difficult to copulate, this meant the 2 varieties of Suturalis were each prevented from mating with their neighboring species.
The critical part of 2 organisms becoming evolutionarily independent of each other is that they don't exchange genes. And that's really what the definition of species is. It's the fact that they cannot mate with each other. Now, this reversal of coil in Partula, one goes left and one goes right, actually promotes reproductive isolation, because when 2 things have opposite coils, they have a reduced likelihood of mating. And so here we have a case where clearly the change in coil is not a neutral character. It has been the result of natural selection favouring individuals that do not mate with the other group.
To confirm this theory, Brian brought back several species of Partula. They are hard to keep alive in captivity, but Vivian has become an expert in the arcane field of snail husbandry.
It's quite tricky, really, but you get used to it. And the snails certainly do like the same sort of treatment. I've a colleague that works for me with the snails, Joyce, and when she goes away, I do her snails, and when I go away, she does mine. And even though we're both used to handling the snails, we do lose some, both of us do, because whether it's our hands or, I don't really know, but they certainly know anyway.
Vivian's gentle touch has saved 3 species of Partula from extinction. They're thriving on porridge oats. But it's thought that 53 other species are in dire need of her attention. So an international campaign to save the snail has swung into action.
This really is a rescue mission. We're going on to the island of Raiatea now. We've just come from the northern island of Taha, where they are extinct. All the Partula seem to have disappeared there through the predator snail Euglandina. And speed is really of the essence. The sooner we get there, the sooner we have any chance of rescuing some of the species.
Dave Clark has spent the last week scouring the Society Islands for Partula, assisted by Roger Klosak from Chicago. Euglandina is spreading at a rate of 1.2 kilometers a year. It seems strange, but to save snails, they're having to move fast.
We're hoping to link up with the local ministry, the Economy Rurale, and Jean-Pierre Mallet, who's been helping us there before. Come on, Roger, you're going to swim this one?
I forgot my trunks today.
Ironically, it was the Department of the Economy Rurale in Polynesia that originally imported Euglandina from Guam. But today, they are a useful ally in the mission to rescue Partula.
Comment ça va?
Ça va, et vous? Bonjour. Bonjour.
Est-ce que vous prospectez dans cette vallée récemment?
Oh, dans cette vallée, je sais qu'il y a...
According to Jean-Pierre, a small population of Partula had escaped Euglandina's invasion of Raiatea and survived in this valley, one of the most remote corners of the island. But last year, the predator turned up here as well.
Merci.
Allez, au revoir.
Au revoir.
Bonne journée.
Even so, Roger Klosak and Dave Clark decided to look for any survivors.
He said that we may be able to find some up the valley. So,
There used to be 33 species of Partula on Raiatea, and on an earlier rescue mission, Dave managed to collect some of these.
Past these beautiful mimosas on the ground.
It's a long way up, but his faint hope is that he can get to the head of the valley before the predator and rescue whatever Partula remain.
There's a good slope here, so...
Good stanza, Freycinetia, huh?
Yeah. Right, I've got a Euglandina. That's quite a fresh one. Right, we'll have to look for Partula shells then. I doubt if there's going to be anything on the trees. It's what we're expecting about Partula on Raiatea, that they're extinct throughout the island now. This is the last possible place that they could have been. We've checked several other areas as well, and it looks like they've all gone. There's plenty of predators around, and where there are predators, there's not really going to be any snails.
With Partula out of the picture, they decided to assess the state of the other snail populations instead.
It's a giant land snail.
How large is that one, Dave?
It's not...
They also wanted to check what Euglandina had been consuming.
I've got a little predator here, a juvenile.
What, a live one?
Live one, yeah.
Oh, right, yeah, bring that up, because we could do with having a look inside that one.
Okay.
See what they've been eating, because... It's a ridiculous situation, because these predator snails are so efficient at what they do. The only thing is they're very efficient at the Partula snails, but not at the giant land snails, which are the real problem and the real reason they were introduced in the first place. Okay, I'll try and carefully and quickly dispatch this snail so we can have a look at what it's been feeding on. We need to know. Oh, there we go. Look, there's a...
discoidal.
There's one of those discoidals, yeah. Ah, so they're not fast enough to outrun them.
Not all the time, huh?
No. I don't know.
There goes that theory.
Yeah, you've still got giant land snails here, but all the endemic ones, all the very important snails that are only found in these islands, are disappearing, and certainly have gone now from Raiatea, 33 species. And anybody who thinks that it's a good idea to introduce the predator snail, people like, I know the locals think that it's a good idea, and people like Mooney Arpin, who suggested introducing them in the first place. I mean, it's a disaster, it really is, and it's just gutting to look around these places and see them all gone.
But the gardeners of Raiatea have a different story to tell.
You know, it was very funny when we arrived here about 12 years ago. My garden was full of that huge snails. And there were so many, I did not know what to do, so I collect them every morning and I had to throw them in the road because I want to protect my flowers and my garden. And a few years later, I suddenly discovered a strange creature. It looked like a snail, but a snail I'd never seen before. Small, but with a huge, long nail with a kind of like tentacle, you know. And I did not know what it was. And one morning I woke up and I saw that small one, I think you call them predator, now I know the name. It was climbing on this one and it was eating that one. So in a way, I thought it was good news because I feel my flowers were safe from that horrible big thing.
In one way, Marie-Isabelle was right. Her flowers were safe. The plague of land snails did die out. But quite why this happened is still a matter for debate.
When you refer back to authorities such as Mead, it's very clear that when a population in a new environment rapidly increases its numbers, it will inevitably have a population crash again.
I'm not sure whether population naturally dies down, unless there is some factor, some natural enemy, either a disease or a parasite or something else is bringing, or a predator bringing it down. The population, I don't think it will go up and down by itself.
Euglandina was welcomed by many islanders as the predator that could rid them of the African land snail. And wherever Dave went, he found it had got there first. His last hope was the island of Huahine, where he'd arranged to meet up with the family of Professor Brian Clark, valuable extra hands in the bid to save Partula.
We were expecting to find 3 species of Partula and we found 1 snail.
Well, I just couldn't believe it getting into Huahine last night and Clark telling me that there were predators here. I was rather hoping that she was joking to begin with, but it turns out that they really are here. She collected a live one and lots of shells, and she has actually got 1 live Partula. You got 1 live one?
We've got 1 live one.
But that was after a thorough search in one of the best areas, so things are pretty bad. Might be best to split into 2 groups. Actually.
Yes, well we could.
So we can cover more area in a shorter period of time, because I think every second's going to count now. And now going into one river valley to have a look for some other snails in a different area. And it's one that people say there are still snails in, but there tends to be a local thing that they think there are lots of snails and they've been there for several months, but it can be that they've actually disappeared from there quite a long time ago.
L'escargot est plein dans toute l'île de Huahine, surtout dans les montagnes.
I've got a connection to one of the pastors in the local church in Maeva, and he knows the necklace makers, so what we're going to try and do is talk to him, see if we can get some people together to go snail searching and see whether we can find any. Joanna? Vous connaissez où se trouve le pasteur à ce moment?
Il est parti en ville pour célébrer la messe à Fari.
He was supposed to be in Maeva, but he's now in Fare doing a service over there. Sometimes it's as hard to try and find people as it is to find snails.
Dave finally caught up with the pastor at the church in Fare, where he was in the middle of taking a service. But when church was over, he and his flock were only too willing to give their advice.
Je suis David Clark à la Parc zoologique de l'Ombre et je cherche pour les petits escargots. C'est vrai, par le prédateur, il mange les escargots, oui. Les coquillages de terre seulement, les aréoles.
Mais il y a un escargot.
Having spoken to the pastor, I think that he's going to get a couple of people to help us look in a minute after he's done the next service. That could be very useful going into the forest with the local people. They are incredibly helpful, so it's quite useful to talk to them. But I still don't know whether we're going to have much luck, particularly looking behind Fare, because that's an area where the predator has been found. Fare ici. Oui?
The help that the pastor eventually mustered consisted of 4 local children promised a free lunch in return for their efforts.
We managed to get quite a few of the species Partula varia, but only managed to get 8 of the other species, Partula arguta, which was one that we were after because there are none in captivity. Ah, bon, merci.
At the end of the day, Dave packed up the few snails they had collected and took them down to the harbour in Fare.
It certainly looks like Euglandina arrived at Fare. They probably came just accidentally. I don't think anybody would have really bothered to introduce them on purpose here because there was the crash in the population of giant land snails anyway. Unfortunately, as they are here, it now means that all the species will soon be extinct in the wild. So we're lucky to get what we can. I got those packaged up, ready to send back to Britain. And the only chance now for any of the species here is captive breeding, because they'll be completely extirpated from the wild.
And so Partula, plucked from the last place they might have survived in the wild, was sent through the post to that final resting place of so many of the world's endangered animals, the zoo. This is where the husbandry and the breeding programs are being developed that might allow not just Partula, but hundreds of the world's threatened animals to ride out the storm of late 20th century existence. But the techniques and the successes of the Partula program are testing the usual approach to captive breeding. The scientist in charge is Georgina Mace from the Institute of Zoology.
When we set up a captive breeding program for a new species, we want to make sure that the population we have is at least remaining stable. And that means looking out for 3 particular aspects of how we keep them. The first one we would call basic husbandry, which includes the diet that we're feeding them. Is it the right diet for them? The second one is disease. Clearly, if there's some pathogen or disease in the population, then the population's not going to sustain itself. The final one is that we need to maintain genetic variability within the species. And one of the ways that we maintain variability is by minimizing the opportunity for individuals to inbreed. That is, to mate with their close relatives.
For this reason, most captive animals are outbred as vigorously as possible.
In fact, it's more complicated than that even, in that those big outcross populations, because they always contain lots of genetic variability, are the ones that are most rapidly going to adapt to captive conditions.
So because either kind of breeding has drawbacks, Georgina Mace decided to take a risk with Partula. She did both. She split the population into 2 groups. Half are being outbred in the conventional way to maintain genetic variability. The live-born babies are taken away from their parents and put in to reproduce with snails of the same species, but from other places. As time goes by, these snails may lose the genes that allow them to survive in the wild. The other group are left to reproduce with their relatives. The idea is that successive generations will keep the genes that allow their wild existence, though at a cost. There is a risk that they will suffer the effects of inbreeding. But the computer analysis of the survival of both groups over the past 3 years shows something rather alarming. The red line shows the crossed outbred populations, and the blue the inbred.
Well, what's clear is that the inbred lines have died out. The crossed lines have been doing much better, and have, in many cases, been increasing in number quite radically. Now, obviously, that means that we're maintaining a good population, but we're worried that we may gradually be having a population of snails that's just adapted to captive conditions rather than to the wild.
So, from glass tank to glass house, outbred Partula were shifted 10 miles across London to Kew to see if they could withstand conditions close to their wild environment. Georgina Mace was convinced there would be a lot of deaths, but the liberated snails not only went forth, they multiplied.
There were actually no mortalities and 2 births, which we were really surprised by.
What led to this success might be very straightforward.
One possibility is that snails have a few very simple rules that govern their behavior, and that those are probably genetically determined. So, for example, you could have a very simple rule: if it's sunny, go under the leaf till you're upside down. All right, then.
This sensible behavior shows that outbred Partula have kept their ability to survive in the wild, an ability that made possible the next and most exciting stage of the captive breeding program, the return of the snails to Polynesia. Such a major step required the gathering of what is known as the core group of the Partula program. Vivian Frame and Brian Clark took the early train down from Nottingham. Quentin Bloxham came from Jersey. The rest just came from their various duties in the zoo. They all converged on the Conservation and Consultancy Division of the Zoological Society in Regent's Park. The first item on the agenda was the location of the release site.
The release is there's a little road that goes up the centre of Moorea here, and it goes up to a belvedere, which is a lookout point. And then you take off that road sideways, already at quite high altitude, and there are some very good clumps of climbing pandanus there. And we know that they're good for snails because we collected masses of snails there before they went extinct. And Jim has done the selection of appropriate places for release, so it's handy. You can get up there with a vehicle, and it's a good part of the habitat. And it's actually a place where Tahitiana used to live.
But with the location selected and the foundations being dug, the core group still needed to decide on a way of keeping the predator out, the subject of much reminiscence by Brian Clark.
Which leads me to think that we may have to go back to our first idea, which is an electric fence, anti-snail fence. That worked a treat when I was doing it during my defill. It would keep the snails in wonderfully because they would crawl across and we get 12-volt shot, froth like mad and fall off.
In the end, the design for the reserve's fences included a combination of 2 anti-Euglandina features.
The walls of the enclosure are made of these sheets of corrugated galvanized iron. They will keep the Partula on the inside and the predator on the outside. Our first line of defense against the predator, however, which could easily climb over the galvanized iron, is this trough, which will contain a mixture of calcium chloride in a toweling substrate. And we've shown that that is an inhibitor for the predator. Should they, however, get over that one, then the next barrier is along this strip of wood here, where we are putting 2 lines of electrical wire. We will also have to wire the large branches that arc over into the area, which might form bridges whereby they could cross into the area.
But even if these elaborate defenses cannot be breached, there is the danger that the enemy may already be within.
It's a dead Euglandina shell.
A thorough search was conducted for Euglandina, and as an extra precaution, all the leaf litter which might be concealing eggs was removed from the site. And with Euglandina out, the next decision was which Partula to bring in.
Viv, you're going to be bringing down?
Sutralis and Turbiana.
And you're going to be getting them ready, of course, aren't you?
Yes, we're getting ready. And we're bringing them back down. This is about 22 boxes.
Right. And we're also going to have Chester,
yep,
will be providing, and also Edinburgh, of course,
right,
and also Martinmere. So coming together.
The decisions all taken, the core group disbanded, secure in the knowledge that they had covered every eventuality. Now the priority was the transportation of Partula, a task allocated to the safe hands of Vivian Frame.
Each of the snails put in the middle of a tiny piece of toilet roll and closely folded up, and they'll just sleep. We'll pack 10 probably in each box, and then they'll go to Paul in London Zoo, who'll take them all to Moorea. There'll be about 30 boxes, these white boxes, packed in a bigger box. I'd be silly if I said I wasn't worried about them going, but I do feel they have to have the chance, and this is the place they should be at anyway, not in a lab.
The snails were carefully boxed up and sent on the first leg of their journey down to London, where Paul Pierce Kelly was doing some Polynesian packing of his own.
I'm very excited indeed about getting the snails back out to Polynesia again. It really does mark a very important milestone in what's been a very long international effort to keep the little things going until we can begin the release stage.
There's a good one. There's a... Right, say bye-bye, Daddy. Bye-bye.
But this international effort and this international travel may all be in vain. For while Paul, the Partula, and his wife Alison set off for Heathrow, on Guam, Dr. Munyapin is researching a further method of biological control. Predator 2 is the New Guinea flatworm.
Flatworm is parasitic of the snail, and when the snail moves and it leaves a slime on the ground, the flatworm follows the slime and then it reaches the snail, and once this gets on the body of the snail snail tries to retract the body into the shell. In that process, it takes the parasites also inside, and the parasite feeds on some of the vital organs, and the snail dies.
As far as the introduction of the New Guinea flatworm is concerned, the risks are probably of several orders of magnitude greater in terms of guaranteed species loss. Wherever they are introduced to biosphere, you know that these things are guaranteed going to result in the eradication of pretty much 100% of your endemic snail fauna.
As the Partula continue their journey home, the New Guinea flatworm has already reached the Philippines and the Maldives. There are no plans to send it to Moorea, but there is always the danger of an accidental introduction.
What I feel about Dr. Munyapin, it's difficult to put into words, and without wishing to go into the realm of being personal, I would just say that it has been said that Dr. Munyapin is a strong candidate for being responsible for the greatest number of documented species losses in history.
I don't think it's true to say that people just don't care about the fate of something like these little animals. It's probably true to say that many people aren't aware, and consequently you cannot be concerned about something of which you do not know. But they're two very different things. They are as endangered as you can possibly get, in as much as they are now completely extinct from the wild range.
After their journey of 10,000 miles, and despite the threat of 2 lethal predators, Partula are finally home. Deep in the Moorean forest, their safe haven awaits them.
Oh my God, this is incredible.
Up to the top, up to the top.
It's amazing.
Here go the babies.
Here.
Oh, put them right in here.
Wow, what a lot of work. Okay, little ones, not far now. Another tragedy would be if the flatworm got into the Society Islands. Okay, to have another predator that we have to deal with as well would just make things so much more complicated. But we've got this one little oasis for them, which hopefully will found a nice little population.
Oh, it's absolutely marvelous, isn't it? To be able to put Partula back in the wild for the first time in years. It's wonderful.
Right, we'll do.
I'll put some on this Angiopteris here.
Careful not to crush the veg.
Yes, do be careful. We've been working on Partula since 1962. Of course, it was a great shock when we learned about the final extinction. And what we hope to do is to get some good science out of this disaster.
And what we're doing here is testing our earlier hypotheses that the major factor, one of the major factors in the evolution of Partula, is the other species that are present. And we're putting one species in each of 3 of these quadrats and 3 species in the 4th quadrat, and we can see when they're put together how they interact with each other. So here we're making not only a conservation effort, but an experiment in science to test our earlier hypotheses.
This is tremendously exciting because although there's very real science going on here, the conservation spin-off, of course, has been tremendous. And we are now at the stage where there are Partulas back in their natural habitat.
I like it.
That looks like a Nottingham one to me, Viv. In it goes. This is as natural as you can possibly get. If we see the Serengeti and the wildebeest, that is managed. There's no difference at all. The difference is the size of reserve is smaller in this case. This is a reserve which, as it happens, is for snails.
Really amazed. I didn't think it'd be anywhere near as big as this. I think we've done really, really well. People have worked really hard, and it was just amazing. The fact it's actually all come off, it's all happened. But they shouldn't be living in labs and little lunch boxes. This is where they should be.
One can never regenerate the variety of species that made up the original natural populations. What we're trying to do is do the best we can with what we have left.
Someone's got to worry about snails. It's not just the big animals like the pandas and the antelopes that need protecting. It is the small, slimy creatures as well.
Returning Partula to Moorea may be a sort of symbolic act in some way. At least we've tried to save some species, because when you lose a species, you can never get it back.
Well, whether Partula have a future or not is, I think the jury is still out. But thanks to a tremendous international effort, they do have a chance. And I think a very real chance.